Good afternoon, ladies and gentlemen, and very nice to see you here. My name is Aussara from Faculty of Science in my university, and I would like to introduce myself a little bit more. So now in Chiang my university, we try to establish our research group, which is called Cell Engineering for Cancer Therapy. So I and my team, mainly we produce all kinds of immunotherapy, which are very strong collaboration with Professor Peptide here. So in our lab, actually, we have a dendritic cells technology, we have a CAR T cells, we have the CAR NK, we have bispecific T cells engager, bispecific NK cell engager, and what I recently published, and I select this to be my topic today is a peptide vaccine, but it's quite a little bit different from Thailand because we designed from the information of driver genes mutations. So it's a shared common antigen. So this is what I'm going to talk today. So, or I think briefly, I have only 20 or maybe 30 minutes to give my presentation. So if you cannot catch up my talk, or if you have other comments and questions, please feel free to email to me. We'll be very happy to answer to your question. And this is my result just published last year, the end of last year. And we do design peptide vaccines and test it in vitro in the models of cholangiocarcinoma, and this is we publish in Biomedicine and Pharmacotherapy. So this is our result in this paper. And I would like to give a brief talk about our disease model, which is a cholangiocarcinoma. I will make it called the chart with the CCA. So this bile duct cancer actually is a rare disease worldwide, but they have high prevalence in Thailand, especially in the northeast of Thailand. And a couple months ago, we went to Konkan and found the information that it's not just only the north is of Thailand, but northern part of Thailand, like in Phra province, still have a high prevalence of the CCA. So what's important here of the CCA is the CCA in Thailand is quite very different and unique from the other country because it's related to the liver fruit infection. So in our countries, it's kind of, we have a different causative etiologies of the cancer. One scary thing is about this disease model because it have low survival rate. You can see it's right here. Actually, most of the patients, about 70% of the patients cannot do the surgery because they have late diagnosed. When they come to see a doctor, they got like advanced disease. So the best way for the treatment is just the systemic treatments like radiotherapies and chemotherapy. And these cancers, this kind of cancers, highly resistant to chemotherapy. This is why the survival rate for this group of patients is just less than 12 months. So even the group that can do like surgery, the survival rate is still low because the kind of nature of this cancer is pretty much recurrent. So if you comparing this bile duct cancer with the other cancer in terms of survival rate, this cancer is still low. This is supporting one thing that the standard treatments of the CCA is not that good for prolong the survival rate. This is why alternative effective treatment is necessary. And I think the best way we could imagine about this is to cure the disease, but somehow, at least if we can improve the qualities of life, or maybe help the people to live longer, it makes sense to fight for. And this is my disease model. And several years ago, I have read this paper. This is belonging to Cholangiocarcinoma Research Institute and Professor Sopit Wongkam and collaborators. So this paper tried to, understand comparing the mutations, comparing between the CCA patient without obesity infections, and this is from Thailand with liver fruit infections. And you will see here that the list of the mutations are quite different between these two groups. So this paper tried to, conclude that maybe the distinct of the somatic mutations it depends on the causative etiology. So what does it mean? It means that if we have the information from the Western countries, maybe lots of patients, we cannot apply it to our patients in our country. One thing we have to think about is if you want to be something for the CCA patients in Thailand, is maybe have to begin with our own information. This is the real treatments for Thai people. This is what's in my mind. I just try to think about how to combine the informations and to build like a unique immunotherapy for the CCA patients in Thailand. So you might know that we have different kinds of immunotherapy nowadays. And what have fit to the informations are select the peptide vaccines. And because if we have a informations of the mutation. So we can use this information to design the peptide, which we call the neoantigens, like Ajahn Tairak have said about. And this because it is neoantigens. So we expect that we have a highly immunogenic and highly specific to only the cancer cell. So you might expect to see like highly immune response and highly safety to use it. And this is what I decided to dig in deeper for the peptide vaccine status nowadays. This is the process. I think Ajahn have talked about this already, but I want you to pin up something that the marketing for the peptide vaccine actually comparing to another immunotherapy, and actually it's quite huge. I mean, this have the room for the peptide vaccine and increases every year. I mean, continuously. But what we have to know about the peptide vaccines that the peptide vaccine nowadays is the personalized medicine. So if you think about the cost effective or not, I think it's not if you're comparing to the technology what we call the off-the-shelf technology. So the cost effective is one thing. And one thing you have to know about the peptide vaccine is therapeutic vaccine. So it is for the treatment, not for the protection. This is to like guide me to do something. I've been a little bit out of the box. So I expect that maybe I can use the informations of the CCA mutations, try to find something what we call the mutation hotspots in the landscape, and find out the common antigen, common neoantigen. After that, I can predict it. I mean, by using bioinformatics. And we design the neoantigen peptides. After that, we can induce this peptide, I mean, challenge to dendritic cells and use dendritic cell to activate the T cells. And finally, in this project, I try to like understand and test the efficiency of the T cells to kill the cancer. That is what the things happens in this paper. So I'll begin with this one. So I'm beginning to try to discover what we call the chair antigens, so the chair neoantigen. So what I have in my hands, in that time, I have only the cell line. So we have a cell lines in our laboratory, which is established from the Thai CCA patients. So I think let's begin with this. So I try to do the whole exome sequencing. And at that time, I expect to see something, what we call the hotspot. But unfortunately, I cannot find one. It seems that the mutations it says first throughout the exome. So I cannot find a hotspot, except as though I sit back and talk to myself. Maybe I have to begin in what we have already knows about. So I try to map what I have about mutations to what have been reported recently. So I map it back with the genes which is have been recently, I mean, reported before. So I'm looking for the mutations in these 19 genes, which is published from the PIT group. And finally, I'll find something. I mean, likely I find some point mutation which is shared between the cancer. And this is, you can predict it. It's the driver genes. So it has some driver genes as can in common, but not that much. I select just only three things to begin to design the peptide vaccine. So I select P53, the KRAS, and RNF43, which is the genes related to the CCAs from Thai patients. So I use this information, and the thing is that we use the information of mutations and predict it with the bioinformatics. And I try to design what we call the neoantigen peptide, which is a short peptide, because I want to design these peptides to specific to bind to HLA antigen. So I put the short peptide and hope it can bind to the HLA on the cell surface. And we select the HLA 11, which is high prevalence. I mean, that's the high frequency of the HLA type from the Thai population. So I use these information to decide the peptide, synthesize the peptides, and after that, I test that the peptides can actually bind to the HLA-11 by use the computational docking. So we use molecular docking to find it out. So this is a picture which is present the peptides can sit on the HLA molecules right here. And after that, we try to test it in vitro that actually the peptides can bind to the HLA-11 or not. So we use the T2 binding assay. These specific T2 cells have only the HLA-11. So after the bindings of the peptides on the HLA molecule, this can stabilize the HLA on the cell surface. So you can see that after I put the peptide, this can stabilize the peptide on the cell surface. So I'm pretty sure that's have there on the HLA molecules on the cells. And after that, we try to check with ELISpot so these peptides can activate the immune response. So this is the first beginning. Because in my lab, we have a dendritic cell technology. So we produce the dendritic cell from the monocyte. We call it monocyte-derived dendritic cell. So I think what I try to do is because I have dendritic cells in my hands, so why not I put the peptides directly to the dendritic cell? Because 100 sure does it have, I mean, meat in the well. So we put the peptides to the dendritic cells and hope the peptides can sit on the HLA molecules on the dendritic cells. So I test that the dendritic cell have something, I mean, any problem with the differentiations of dendritic cells or not. And this is a picture. It seems like the dendritic cells just go differentiations perfectly, even we have the peptide. So I try to track it down. So what the benefit if we can induce peptide. So it seems like the other co-stimulatory markers of the dendritic cells markers not that change, except these two, CD or AT and ATC. It seems like when you have a peptide in the system, it's have highly expression of these CD, AT and AT6. It means that the dendritic cell have more like a little bit because these two things is important for the interaction between the DC and T cell. So I hopefully that with the peptides, it have like improved function to present these peptide to the T cell. So we do after this we do co-cultures about the dendritic cells and the T cell, and we test in vitro that the T cell which is activated by our peptide can kill the cancer or not. So this is the way which we use for the killing assay. So I engineered the cancer cell to have a red fluorescent protein. So the cancer cell have in red, and this is little dot is a T cell. So this is how we can track which one is the T cells and all the cancer. So after co-culture 24 hours, so we see how we have the cancer we have left in the wells. And what I have done is at the beginning, this is a tested cell. So I select KKU213A, which is carrying the HLA-1101, which is we designed for. And this is the first batch of my killing assay. So you can look up here. This is a control cell. We have these numbers of the cancer. But after we put the T cell, which is activated already with the dendritic cells, so you can see that we have a lower numbers of the cancer cell. And this is a positive control. We use a tumor lysis to house the dendritic cell. And this is how the peptide works. So this is the KRAS, that's a peptide from NIF 43s, and this is a P53, and this is a pool peptide, which is have a combinations of these peptides together. And it seems like the peptides can actually activate the T cells and the T cells can do function to improve the killing assay, and we confirm it with the determinations of the granulolysis and granzyme A, which is confirmed that this T cell is have improved in healing's activity. So to test back that this is specific killings or not. So we use the same T cells and test it with the KKU 100, which is not carry the 11 HLA-11, but instead it have the HLA or 33. So it seems like when we put the same T cells to this cancer cell, it's have nothing happen. And this is we can we can confirm that the killing activities of the T cell is restricted to the HLA-11, which we decided the first. So if we have not matched HLA, I mean molecule, so it cannot kill the other things. So this is what I have done for this paper. What I have a little bit more discuss with you is 2 points of this my study. The first one is the peptide vaccine efficiencies among the individuals. What we have, what we have seen in the last slides is that it's a pool result from many donors. But if you look carefully, it's, for example, for these 3 donors, you will see that the tumor death is quite very different in these 3 donors. Particularly if you're looking carefully in donor 3, you will see that the peptides cannot, individual peptides somehow it cannot like in some ones it cannot do like a very equal efficiency. It's because of what? I think it's because of the T cells repertoires of each individual. So in the terms of peptide vaccine, I think the combinations of peptides is maybe, important to induce the immune response. For the T cell repertoire, which is maybe different among individual. And this is the one factor which is affect to the peptide vaccine. Another thing is immune status. So if you thinking about the patients, a healthy donor, and yes, the immune response of the patients may be a little bit low. So this is the factors we have to concern. And the factors I call it the quality factor, which is you have to have a T cell repertoire which is respond to the peptides, and immune status is very important. The second thing is what I call it the quantity factors. So the peptide vaccine nowadays is therapeutic vaccine. So we don't have like the protective vaccine. So I just thinking about how, just imagine to the situation that we could have a protective vaccines, and thinking about what's the drawbacks or maybe the limitation. So one thing I just thinking about is how can we sure it's the immune response from the peptide vaccines lasts that long. So I try to put this result from the NeoVax clinical trial to use the peptide vaccines. It sounds like the anti-tumor response is persist over the years. So if you're thinking about this last that long, I think the peptide vaccine is something that can have that last long. So I just thinking that apart from the therapeutic vaccines, if we can do protective vaccine for some cancer, which is, that the areas have the high risk of the cancer, just like the CCA in Thailand. We know that the high prevalence in northeast of Thailand, north part of Thailand, and is heavily related to liver infections. So what's about protective vaccine for these people that we know that is have a high risk to be onset for the CCA? I think this is a thing I try to think and try to discuss in this paper. I think for protective vaccines versus therapeutic vaccine for the CCA, what is or not? I think you have the answers and have mine. So this is the one thing I would like to share with you. So before I finish my presentations, I think I have to give like many thanks to many people to make this happen. Professor Mahidol, who always support me, my friends Sutama, Mr. Hatai, which has helped me a lot to do about different things I tried to do. My friend Professor George Bailey from University of Glasgow, and for today it's not from our team, not just only me to give the presentations. And today, I think my postdoc will give the presentation on trispecific NK cell engager, and I hope you enjoy it. And this is my last slide. So I would like to say thank you with Kumamon. And I cannot say it in Japan, but I love this slide. So I would like to say thank you, and I would like to say thank you to the Harvard Talents who, let me to be here. I very happy to be here and share with you all of my things inside the lab. And if you have any questions, I'm here, and very happy to take the questions and answer if I can. So thank you very much.
Associate Professor Dr. Aussara Panya for giving us a talk on interesting topic. So, is there any questions from the floor?
Hi, that was a really nice talk. I was just wondering about the fact that you mentioned on the protective vaccine, which is a great challenge to be honest. So would you like to comment on that? Because for cholangiocarcinoma cases in Thailand are majority caused by liver variant, like you said. So it's unlike other cancers that may be caused by unique cellular organisms or bacterial infection, for example. So what could be further steps that, if someone would like to take that path? Yeah.
To for protective vaccines, right?
Yeah.
I think that it's not only the researcher to make a move on this. I think there's a policies and things. So we know we know the problems in Thailand. We know the CCA. We know the high risk. It's not this of Thailand. It's not part of Thailand. But to apply the vaccines in all local peoples in that area is impossible. I know that. I think it's we if we want to gear up this policies, we have to work with the I mean government unit to I mean we have to set up the regiments to who uh that who's that the people who can get this vaccine. So we have to find the criteria so that these people have high risk and I mean worth enough to have the vaccine because the if we call the vaccines, even the peptide vaccine is have a cost. So it's not that cheap. But I think if we can prevent the cancer, it's something it's worth to do because for the cancer treatments it's not that small numbers of money. That's a lot. So if we can make it happen in the real, I mean, like have a cohort study, if it sounds now, I think it will change a lot about CCAs in Thailand because the CCA have a long onset. It's quite like 20 years. You cannot predict another 20 years. Do you have the patients with the CCA or not? People believe that the CCA will be disappear one day because you have a very good education. But once we have that's one that we went to Khon Kaen University and a director said that what we have here is the result from 20 years ago. So you cannot predict that what's the art of the 20 years in the future. Do you have can CCA disappear or not? This maybe not. So I think it's worth to thinking about this and make a move about this.
Thank you.
ค่ะอาจารย์
I think you might already aware that literature that report that those oncogenic mutation, those struggle, especially it's not only the cancer cell that escape from that.
Yeah.
Right. So have you look at the tissue sample from the real patient? Are those truncal mutations still expressed?
I understand. I understand your point because many people will be asking if it's work like driver mutations can have a good immunity, why is the cancer still happen? This is a big short of the questions. But as I said that in my opinions, it's having two terms in quality and quantity. If we use like a treatment, maybe it's not sufficient enough. I mean, when people ask me, do the immunity is exist? I think yes, but is it sufficient to stop the cancer? I think maybe not. Because of what? Because we put the peptide follow the cancer. So I think that if we put back, we try to protective vaccines, we get ready to have these. Maybe we have both in qualities and quantity to protect it. Often this is my idea, but I'm not sure it's good enough to take a short on protective or not. But this is one thing I think it's not just a quality. The number is important. So they have to be like sufficient to stop all of the cancer to be happened and before it happened.